You know, when most people think about brewing beer, they have this really romantic image in their head. I'm guilty of it too.
Oh, definitely.
You picture a guy with a beard, flannel shirt, stirring a big steaming pot with a wooden paddle. It's all very, pastoral.
It's a lovely image. It sells a lot of beer, feels traditional, handmade.
Exactly. But after spending the last few days with the source material for today's deep dive, brewing science and practice, well, I think we need to shatter that image.
Or at least add some safety goggles and a lab coat to it.
Right. Because what we're diving into today is this realization that brewing is, at its core, a really rigorous, high-stakes branch of chemical engineering and biology.
It absolutely is. I mean,
Oh, yeah.
this text we're looking at is not a recipe book. It's a molecular deconstruction of how beer even comes to exist.
It's intense.
We're not just talking about add hops for flavor. We're talking about everything from, like, the microscopic anatomy of a barley kernel all the way to the mineral composition of water.
So our mission today is to peel back that label. We want to understand the why behind the beer styles you see on the shelf.
But why?
Yeah, why a stout tastes like coffee, why a pilsner needs a very specific type of water, and why modification is maybe the most important word a brewer knows.
And that word, modification, that is the perfect place to start. But to get there, we have to look at the absolute foundation of the whole thing,
The grain.
the grain, specifically barley.
Okay, so Hordeum vulgare,
Yeah.
the king of brewing grains. My first question is, why? Why? Barley. We grow lots of grains, wheat, corn. I always just assumed it was about the flavor.
Flavor's part of it, for sure.
Yeah.
But the sources point out that it's actually more about mechanics. Right, the architecture of the kernel itself. One of the most critical details is the husk, the parts called the lemma and the palea.
The lemon and the palea.
Right, and in barley, that husk sticks on tight. Even when you thresh it, it stays on.
Okay.
So later, when you grind that malt and mix it with water in the brewery, the husk stays pretty much intact.
And that matters because? Why?
Because when you need to separate the liquid sugar, the wort, from the solids, those husks settle and form a natural filter bed.
Oh, I see. So the grain basically brings its own filtration system with it.
Exactly. Now compare that to wheat. Wheat's in a naked grain. It loses its husk.
So if I try to brew a beer with 100% wheat,
You are going to have a very bad day. You get this gummy, sticky mess. There's no husk, no filter bed, and the whole thing turns into something like cement.
a stuck mash. I read that term.
It's a brewer's nightmare. So barley is chosen because it's mechanically cooperative.
Okay, that makes sense. But we can't just brew with it straight from the field. We have to turn it into malt.
Right, and this is a huge aha moment for me. You don't grow malt. Malt is something you make from barley.
And the way you make it is, well, it's a little weird when you think about it. It's kind of like controlled spoilage, isn't it?
Controlled growth is the polite term.
Right.
You're tricking the grain into thinking it's time to grow. You give it water, warmth, and then right when it produces all the good stuff you need, you kill it.
Ruthless. Okay, let's break down this controlled growth. The text says 3 steps: steeping, germination, and kilning. Steeping seems simple, just soaking the grain.
It sounds simple, but it's a real balancing act. You can't just dump barley in water and walk away. The grain is alive. It's dormant, but it's alive.
It needs to breathe.
Exactly. It needs to breathe.
Yeah.
If you leave it underwater too long, it literally drowns.
I did not know you could drown a seed.
You absolutely can. So the text talks about air rests. You soak it, then you drain the water and pull fresh air through the grain to get rid of CO2 and give it oxygen. Then you soak it again.
You're trying to wake it up without suffocating it?
Precisely.
Okay, so once it's awake and hydrated, we get to step 2, germination. And here's that magic word, modification. What is actually happening here?
So this is the biological phase. Think of the barley kernel as a fortress. Inside you have all this starch, your potential alcohol, but it's locked away in cells with tough walls made of beta-glucans and protein.
Like a locked treasure chest.
Perfect analogy. And the brewer doesn't have the key. The embryo inside the grain has the key.
Okay.
When it starts to germinate, the embryo releases hormones, gibberellins. These hormones trigger the production of enzymes.
So the grain makes its own tools to break down the walls.
Exactly. The enzymes attack the cell walls and the protein. They dissolve them. And that process of breaking down the walls to get to the starch, that is modification.
And if you don't do it right,
You get what are called hard ends. The text describes a rub-out test, which is such a great tactile thing.
I loved this detail.
A maltster just grabs a grain and rubs it between their finger and thumb.
And if it's well modified,
It crushes into a fine powder, the structure's gone. But if you feel hard, gritty bits,
Mm.
those are the hard ends. That's unmodified grain. That's bad.
And there's a visual cue, too, the across spire.
Yep. That's the shoot of the new plant.
Huh.
Grows into the husk, so you can't really see it unless you peel the husk back. The length of that shoot is like a progress bar.
Yeah.
Tells you how far along the breakdown is.
So the green has sprouted, the enzymes are going wild, the cell walls are mush. We have green malt.
Which isn't actually green, by the way. It just means immature, like green wood.
Right. But we can't brew with this yet. It's wet and it probably tastes like grass. We have to stop the growth.
And this is where we get to kilning. We move from biology to, I guess, thermodynamics. We apply heat. And the way we apply that heat determines almost everything.
It's like painting with heat.
It really is.
Yeah.
But you have a big dilemma. You need those enzymes to survive for the brewery. But heat kills enzymes.
So for a pale malt, for like a lager or a pale ale, you have to be really careful.
Very gentle. You cure it at lower temps, maybe 80 degrees Celsius. You save the enzymes, but you don't get much color.
But then you have the dark malts, the ones that taste like toast or caramel. That's the Maillard reaction rate. I usually think of that with searing a steak.
It's the exact same chemical magic. Sugars reacting with amino acids under heat. They create compounds called melanoidins, and you get all these flavors, nutty, bready, roasty.
And the text makes a big distinction between crystal malts and roasted malts. I never realized they were made so differently.
Oh, they are worlds apart. With crystal malts, you do something, well, it's pretty counterintuitive.
What's that?
You take that wet green malt and you don't dry it, you stew it first.
Stew it.
Yeah, you heat it up to about 65, 75 degrees Celsius while it's still moist. You're basically mashing inside the husk.
So you're turning the starch into sugar right there in the kernel.
Exactly. The enzymes liquefy the starch inside the grain. Then you crank up the heat and dry it, and that liquid sugar cools into a glassy, toffee-like mass.
Wow. Okay, now contrast that with roasted malts, like chocolate or black malt for stouts.
Right. For those, there's no stewing. You're roasting dry grain, like coffee beans, in a huge drum at really high temps.
How high are we talking?
Up to 225, 230 degrees Celsius.
The source material said that's actually dangerous.
Oh, fire risk is a very real thing. You're pushing dry organic matter to the edge of combustion. At those temperatures, you kill everything. No enzymes left. The flavor becomes burnt, dry, astringent.
So you have to be careful not to make your beer taste like an ashtray.
Precisely. It's all about control.
Okay, so we've got our malt. But modern brewing isn't always 100% malt. We have to talk about adjuncts.
The A word. The great debate.
Right. You know the purist stance. The German Reinheitsgebot. No corn, no rice. But the text makes a strong case that adjuncts aren't just about being cheap.
No, and that's a huge misconception. An adjunct is just any source of sugar that isn't malt. And yes, they're often cheaper. But there are big technical reasons to use them.
Like nitrogen dilution? Can you break that down?
Sure, so sometimes barley malt has too much protein. It's high nitrogen. Too much protein can make your beer hazy or cloudy.
So you add something like corn or rice to thin it out?
Exactly. Corn and rice are almost pure starch with very little protein. So you dilute the overall protein level. You get a clearer, more stable beer with a lighter, crisper body. It's an engineering choice.
But you can't just toss raw corn in with the malt, right? There's a problem.
The gelatinization hurdle.
That part was dense, so starch is like a tightly wound ball of string.
Good analogy. And enzymes can't work on it until it unwinds. That unwinding is gelatinization.
And for barley, that happens at a pretty low temperature.
Right around 60 to 62 degrees Celsius, which is perfect because that's where the malt enzymes are happiest.
So it all works in harmony.
But corn and rice are stubborn. Corn needs to get up to 77 degrees. Rice, even higher.
And if you heat the whole mash that high...
You kill the enzymes. They denature. So you're stuck. You've got unwound starch, but no enzymes left to convert it.
So what do? The text mentioned a cereal cooker.
It's basically a separate pressure cooker. You have to boil the corn or rice on its own to gelatinize it. Turn it into this hot goo, and then pump that goo into the main mash where the enzymes are waiting.
It just adds this whole other layer of industrial process.
It really does.
Which brings us to the biggest ingredient of all, one we never think about. Water.
Or liquor, as it's called in the brew house.
Right, liquor. The text had this great line, geography is destiny.
It's a historical truth. Before we understood water chemistry, you brewed what your local water let you brew. You had no choice.
Let's take the classic examples. Burton-on-Trent in England, famous for IPAs.
The water there is incredibly hard. It flows through gypsum beds, so it's full of calcium sulfate.
And that does what?
It makes hop bitterness pop. It gives it a crisp, dry snap. If you brewed that same beer with soft water, the bitterness would be sort of flabby and dull.
So the water literally changes how you taste the hops.
Drastically. Then you go to the total opposite, Pilsen in the Czech Republic.
Famous for Pilsner lagers.
And their water is incredibly soft, almost no dissolved salts. It lets that soft, delicate malt character shine through. If you use Burton water for a Pilsner, it would be harsh and metallic.
And what about Munich?
Munich water is rich in carbonates, which are alkaline. They raise the pH.
Which is bad for enzymes.
Right. But dark roasted malts are naturally acidic.
Ah, I see where this is going.
The acidity of the dark malt canceled out the alkalinity of the water. It was this perfect accidental chemical match. That's why Munich became famous for dark lagers. The water demanded it.
That's fascinating. But today brewers treat their water, and the text goes into some scary stuff they're treating for, not just flavor.
Nitrates are a big one. From agricultural runoff, fertilizer.
Why are they bad for beer?
They can reduce to nitrites, which are toxic to yeast, so your fermentation stalls. Even worse, in humans, they can form carcinogens, so removing them is a safety issue.
Then there is this acronym THMs, trihalomethanes.
Sounds nasty, doesn't it?
Yeah.
It's a byproduct of sterilization. We use chlorine to kill bacteria in city water. But if that water has organic stuff in it, like decaying leaves, the chlorine reacts and forms THMs.
And they taste bad.
They give you these medicinal plastic-like flavors, and they're also a health hazard. So brewers have to scrub them out with carbon filters. It just shows water is not a blank canvas.
And that actually leads us to the next topic, enzymes. We talked about the natural ones, but the text gets into exogenous enzymes.
Which is just a fancy way of saying enzymes you buy in a jug.
Okay, wait. Isn't that cheating? Why would a brewer do that?
I'd call it insurance, not cheating. Remember those gummy beta-glucans from the barley cell walls?
Right.
If the modification wasn't perfect, they can make your wort thick like concrete.
Concrete is not a word I want to hear.
It gets viscous, it clogs everything, so you add some extra beta-glucanase to break down that gum and keep things flowing.
Okay, that makes sense. Then there was one that sounded like a sci-fi villain. Bacterial alpha amylase.
From Bacillus lycoformis.
What's its superpower?
It loves heat. The malt's natural amylase dies around 75 degrees Celsius, but this bacterial one, it comes from an organism that lives in hot springs. It works at over 90 degrees.
This is what you use in those cereal cookers for the corn and rice.
Exactly. When you're boiling that corn, the natural malt enzymes would be toast. You add this bacterial enzyme to liquefy the starch right in the cooker. It lets brewers do things that were physically impossible before.
It's a total game changer. So with all this tech, how does a brewer even know if their malt is any good? I was fascinated by this one test, the fine coarse difference.
It's a brilliant test. A really clever way to measure modification without a microscope.
Okay, so walk me through it. You grind a malt sample two ways. One is fine, like dust. The other is coarse, like grits.
Right, and you mash them both separately. With the fine dust, you've mechanically destroyed everything. Water gets in, sugar comes out, you get a high number. Easy.
But the coarse grind, that's the real test.
That is the test. Because with the coarse bits, you're relying on the malting process to have done the work. If the malt is well modified, the internal cell walls are already broken down. They're like a sponge.
So the water just soaks right in and pulls the sugar out.
Yep, and the amount of sugar you get from the coarse grind will be almost the same as the fine dust. The difference is small.
But if the malt is poorly modified,
Then those coarse bits are like hard little fortresses. The water can't get in, so you get a tiny amount of sugar from the grit, but a huge amount from the dust. The difference is large.
so a big number is bad news. The maltster failed.
Precisely. It's a single number that tells you exactly what happened inside that kernel weeks ago.
So what's the big takeaway here? We've gone from the tiny growth of a shoot inside a grain to, the geology of entire regions.
I think the takeaway is that beer is a product of extreme balance. It's not just art. You're balancing enzyme activity against heat. You're balancing nitrogen levels against clarity. You're balancing minerals against flavor.
And balancing tradition with modern chemical engineering.
Absolutely. The brewer is just constantly monitoring all these invisible variables to keep the whole system from falling apart.
It definitely changes how I'll look at my next pint. But before we go, I have to ask about one machine. The friabilimeter.
Ah, the friabilimeter.
It sounds like a medieval torture device.
It kind of looks like one. It's basically a metal drum made of wire mesh. You put the malt in, and a heavy rubber roller presses it against the screen as it spins for exactly 8 minutes.
A stress test.
It's the ultimate test of that modification we've been talking about. You
Hmm.
spend all this time pampering the grain just for this one moment. Will it crumble?
And if it doesn't?
If it's hard, if it's steely,
Yeah.
it just stays inside the drum. It fails. And that, I think, is the paradox of brewing we should leave people with.
What's that?
That we build this incredibly complex biological architecture just so we can destroy it as efficiently as possible.
Yeah.
You have to destroy the architecture of the grain perfectly to create the architecture of the drink.
That is a wild thought. So next time you're sipping a beer, you can ask yourself, was the barley crushed or did it crumble?